Welcome to the Science Experience, your source for the most current and impactful research and preprints in science. We're really thrilled to have you join us for another deep dive into the fascinating world of groundbreaking discoveries. If you're as excited about cutting-edge science as we are, please take just a moment to like this video and subscribe to our channel. It truly helps us bring you more of these in-depth explorations, and, well, it supports the Science Experience. Today, we're plunging into a topic that really touches everyone, aging. Specifically, we're looking at a recent study that tackles this profound mystery of why biological age isn't just a number, why it's such a major risk factor for so many human diseases. Our mission in this deep dive is to unpack how scientists are trying to, turn back the clock at a cellular level, not with one, but two powerful strategies, and how combining them might just be the key to extending not just health, but actual lifespan. Get ready for some truly surprising facts here, and maybe a look at what could be a revolutionary shift in how we approach longevity. Okay, so let's unpack this. Imagine your body, not just as a machine, but maybe like a living city, constantly under repair, this continuous maintenance. It's called tissue homeostasis, and it's crucial. We need fresh building blocks, right? And that's where stem cells come in. They're our body's incredible repair crew, basically, ready to replace damaged or lost cells, doing surveillance, replacement. But as we age, this repair crew, it gets tired, their numbers dwindle, efficiency declines.
Precisely. Stem cells are absolutely pivotal for that regeneration. They provide that reservoir of precursor cells. You need them to replace our, fully differentiated working cells that get lost or damaged over time. But then at the opposite end of that spectrum, we have a very different and frankly problematic type of cell. Senescent cells. These are cells that have permanently withdrawn from the cell cycle. They're not dividing, but crucially, they're also not dying off like they should. Instead, they become resistant to apoptosis, this programmed cell death. It's how the body normally clears out damaged cells. What's fascinating here, but also deeply troubling, is that these senescent cells don't just sit there quietly. No, they become biological troublemakers. They start secreting this potent cocktail of pro-inflammatory molecules. This whole process is known as the Senescence-Associated Secretory Phenotype, or SAS, and this SASP directly promotes chronic inflammation. It contributes significantly to age-dependent dysfunction and really to the development of nearly all age-related diseases. So as we age, our stem cell pools deplete while these problematic senescent cells build up. It's like a double whammy for our tissues.
So, okay, it sounds like we have this dual problem then. Fewer capable repair cells and more unruly troublemaker cells actively causing damage. It almost feels like a losing battle as you describe it. What have scientists been trying to do to tackle these issues, maybe individually?
Well, researchers have been pursuing two independent but incredibly promising approaches. The first is cellular reprogramming. This involves taking differentiated cells, cells with a specific job, like a skin cell or a liver cell, and helping them regain more stem cell-like qualities, essentially returning them to a younger, more flexible state. The major breakthrough here involved using what are called Yamanaka factors. That's OCT4, KLF4, SOX2, and CMYNIC, often just abbreviated as OKSM. You can think of epigenetics as like the software telling your genes what to do and when. As we age, this software can get corrupted, leading to cells not working right. Yamanaka factors are like a powerful system reset. They rewrite those instructions to a more youthful state. It shows even mature cells can, in theory, be made young again.
Wow, that's a profound idea, literally hitting a reset button on cellular aging. But I do remember hearing about challenges with this approach, right? Like, continuous application wasn't always good.
That's a crucial nuance. Yes, a very astute point. Early studies found that continuous expression of these Yamanaka factors in mice could actually be quite detrimental. It led to issues like disorganized cell growth, even teratomas, which are benign tumors, and, counterintuitively, even a decreased lifespan in some cases. It really highlights the delicate balance in biology. More is definitely not always better. The key insight that emerged later was that repeated short-term expression was the way to go. That approach turned out to be beneficial. It improved cellular and physiological signs of aging without those harmful side effects. It suggested a very careful, pulsed approach was needed. Now, on the other hand, the second approach is senolytic intervention. This aims to specifically ablate or clear out those problematic senescent cells we talked about. For example, there's a peptide called FoxO4-DRI. It was shown in mice to effectively clear senescent cells. And notably, it also helped them recover from severe cell damage caused by chemotherapy. So that indicated its powerful potential to counteract cellular stress and restore some age-dependent declines. Things like physical performance, fur density, even kidney function improved in those mice. So this raises a really important question. If these two problems, stem cell exhaustion and senescent cell buildup, are interlinked, What happens if you combine these two powerful but distinct strategies? Could they work even better together? Maybe create a truly synergistic effect?
That really is the million-dollar question, isn't it?
Yeah.
The researchers in this deep dive, they hypothesized exactly that, since stem cell exhaustion and cellular senescence likely interact. I mean, you mentioned the SASP from senescent cells can directly contribute to stem cell exhaustion by promoting chronic inflammation. So combining these two interventions could potentially have a truly synergistic effect, maximizing life and health span benefits. And to test this big idea, they applied it to a classic model organism, the fruit fly, Drosophila.
Right. To do this, they developed a pretty sophisticated genetic system in the flies. It allowed them to inducibly express OKSM, those Yamanaka factors, and also a senolytic factor, which they called Sen. The Sen factor was basically a fly-specific version of that mammalian FoxO4-DRI peptide, carefully adapted to work in Drosophila. Their initial findings were quite illuminating, actually, and they really reinforced what we learned from the mouse studies with Yamanaka factors. Continuous expression of OKSM or Sen or the combination. It was actually detrimental to lifespan in the flies. It led to significantly reduced longevity across the board, which highlighted once again the immense importance of precise dosing and timing in these kinds of interventions. It's a good reminder that even the most promising therapies need really careful calibration.
Yeah, it's truly remarkable how often the initial sort of seemingly obvious approach just doesn't pan out in biology. So when they saw those detrimental effects from continuous expression, what was the thinking? What led them to try cycling the treatments instead? That sounds like it must have been a real aha moment.
It absolutely seems like it was. The intuition likely came from understanding that cellular processes often need a delicate on-off switch, not just continuous on activation. So to overcome those downsides of continuous expression, the researchers switched to cycling expression. They activated the treatments for short, specific periods, like just 12 or 24 hours, only twice per week. And that's where the results truly began to shine. That pulsed approach made all the difference.
Okay, and what were those results? Was it what they expected this time?
Well, individually, OKSM expression alone did extend mean lifespan. And interestingly, it even extended maximum lifespan in the female flies, which was significant. C impression alone, senolytic, it increased mean lifespan, but surprisingly, it had no effect on maximum lifespan. This led to that phenomenon called mortality compression, where excess late deaths kind of compensate for the earlier protective effects. It suggests you live longer initially, but maybe the underlying aging process catches up more aggressively later on. But the combined intervention, that was the true game changer.
Right, so it wasn't just additive then. When they combined OKSM and Sen expression, particularly that 12 hours twice per week schedule, what did they actually see?
They observed the flies experiencing substantially longer mean and maximum lifespan. Both were significantly extended. And crucially, this wasn't just additive. The combined treatment resulted in benefits that significantly exceeded either intervention alone. It really showed a true synergistic effect on longevity. Mechanistically looking deeper, they found that all three treatments, OKSM, Sen, and their combination, protected the intestinal stem cell pool in the flies. That's a critical area because the fly gut has high cell turnover, and it's a major site of age-related problems. They also saw a marked absence of senescent cells across all three treatments, even with OKSM alone, which was a bit surprising. It suggests OKSM might also have some kind of xenolytic-like effects, or at least prevent senescence. Furthermore, the senolytic peptide, Sen, led to a decrease in pro-inflammatory cytokines in the gut, like TGF-A and UpD3. This directly links clearing out those senescent cells to reduced inflammation and improve stem cell maintenance. It really indicates a direct beneficial interaction happening at the cellular level. Clearing out the troublemakers helps the repair crew.
Okay, so what does this all mean in the grand scheme of aging? It sounds like the study went beyond just observing how long the flies lived. They performed a much deeper quantitative analysis of age-dependent mortality using something called the Gompertz Makeham law. Can you unpack that a bit? It sounds like it's not just about counting days lived.
Exactly. The Gompertz-Makeham law is a mathematical model. It helps us understand how the risk of dying changes as an organism gets older. Typically, mortality rates increase exponentially with age. This law describes that curve. So, analyzing the data this way reveals if an intervention isn't just extending life, but actually slowing down the fundamental rate at which aging increases mortality risk. It gives you a much deeper understanding of the quality of that lifespan extension. Is it just delaying the inevitable or fundamentally changing the aging trajectory?
And what did that analysis reveal about these different treatments?
Well, this is where it gets really interesting. It highlights the synergy. They found that while the senolytic treatment, Sen alone, dramatically decreased initial mortality, it also came with that significant penalty we mentioned. an accelerated aging rate later on. The flies live longer initially, sure, but then they aged roughly 3 times faster than the control flies. That led to the mortality compression. OKSM, on its own, reduced initial mortality too, but with only a slight acceleration of aging, much less severe. But when they were combined, the picture changed dramatically. The combination of OKSM and Sen, especially that 12-hour induction, decreased initial mortality by an incredible amount, over 330-fold, compared to the wild-type flies. And crucially, it significantly reduced that age acceleration penalty seen with Sen alone. The effects they saw just cannot be explained by the 2 interventions acting independently. It wasn't additive. This synergistic interaction seems to be the reason why the combined treatment improved both maximum and median lifespan so much more significantly than either treatment alone. It created a genuinely more youthful mortality trajectory overall. It's like getting the benefits without that major downside of accelerated aging.
That is truly remarkable. And to the best of our knowledge, this is actually the first study showing such a robust lifespan extension in an otherwise normal animal through controlled expression of Yamanaka factors, right? And even more so when combined with a senolytic. This really suggests a powerful new avenue for anti-aging interventions. It offers the promise of not just a longer life, but perhaps a healthier one too, because as you said previously, many individual interventions didn't really budge maximum lifespan, but this combinatorial approach did, and quite profoundly.
Exactly. And what's also fascinating here is that the study used mammalian versions of these factors, OKSM and Drosophila. The fact that they worked suggests a conserved functionality across species, which certainly boosts the potential for translatability down the line. This research strongly suggests that therapies combining cellular reprogramming, maybe through gene therapy, maybe other methods with senolytic peptides or drugs, could hold really significant synergistic potential for promoting tissue repair and reversing age-related decline simultaneously. The metabolic changes they also observed in gene expression, that points to rich areas for future research too. It shows broad systemic effects, not just changing cell numbers. If we connect this to the bigger picture, it really suggests that by simultaneously replacing lost stem cells and removing harmful senescent cells, we might achieve a much more comprehensive and robust anti-aging effect, truly reversing cellular tissue composition towards a more youthful state. It's like tackling multiple hallmarks of aging at once.
And that wraps up another incredible deep dive into the real forefront of scientific discovery, the idea that we could synergistically combat aging by targeting both cell rejuvenation and senescent cell clearance. It really feels like a potential game changer. Just imagine the possibilities this could open up for human health span and longevity down the road. It really leaves you wondering, doesn't it? What other synergistic combinations are waiting to be discovered in the biological world? Could this be the beginning of a whole new era in anti-aging medicine, moving beyond just managing disease to actively reversing the aging process itself? Thank you so much for joining us on this journey through the latest research. If you found this deep dive as fascinating as we did, please do remember to hit that like button and subscribe to the SANS Science Experience. That way you get more cutting-edge science delivered straight to you. Until next time, keep exploring, keep questioning, and stay curious.